Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in developmental biology we will be talking about early development in birds. To discuss this topic we have with us our subject expert Dr. Charu Dogra Rawat. Dr. Rawat is assistant professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you. So today we'll be talking about early development in birds. So we'll start with the type of egg the bird has, the type of the fertilization it undergoes, the type of cleavage which happens in the bird development, and then we'll follow up the gastrulation or the morphogenetic movements until the new relation starts. So the specific objectives for today's session would be to understand the avian early development, the type of egg, fertilization, cleavage, to understand the formation of hypoblast, and to understand the formation of the primitive streak. So these are some of the structures or some of the membranes which are transiently formed in case of the avian development. So looking at how the fertilization or where the fertilization happens, the fertilization of the chick egg occurs in the oviduct before the albumin and shell are secreted to cover it. So here the diagram is shown where there is an ovary and then there is complete oviduct which is shown going up till the vagina and the cloaca from where the sperm enters. Now it's an interesting fact that we usually, the developmental biology studies started in chick, and in hen there is only a single ovary because the sperm has to move so much of a distance and oviduct is pretty large. Therefore, only the single ovary is there in case of hen. So the mature ovum, the mature egg is released by the ovary during ovulation and is then incorporated in the funnel shaped opening, the infundibulum of the oviduct. So the infundibulum is the region, that funnel shaped region where it is shown, where the fertilization occurs. The fertilization by the sperm of the cock can occur at the location during a short time period, approximately in the first 15 minutes. Therefore, the sperm cells have to pass the whole way through the oviduct from the cloaca up to the infundibulum for the fertilization to happen. The next region, the fertilized oocyte, starts immediately with the cleaving process and a small germ layer is formed. We will follow this mechanism or follow this process. But right now we have to understand that after the oocyte has been fertilized, it moves then goes to the magnum area where it is loaded with albumin. The two semi-permeable shell membranes are formed at the isthmus, and while the poriferous calcified shell is set in the uterus, the egg is laid around 24 to 28 hours after the fertilization. So the fertilization occurs in the infundibulum region. In the magnum region, the ovum receives the first layer of albumin or the egg white, which is called as, and in the isthmus, the egg and the shell membranes and more albumin is then added. And then you can see there is a shell gland where the final albumin, shell and the pigments are added here, and there are certain papilla which move it through the vagina and then it is laid after the 24 to 28 hours of fertilization. A light contraction of the egg mass takes place after cooling down and results in the formation of an air chamber between the two aforementioned shell membranes. So there is an air gap that exists, and that is because of the cooling down of the egg while passing through the entire area, and therefore where the shell membranes just separate out, they contract a little bit. So this is how the fertilization happens and the journey of the egg from the infundibulum or from the place where it is released to the where it is laid from. Now looking at the type of egg the avians have, the birds have megalethal egg, that is they have large amount of yolk, and a telolethal egg where yolk is usually concentrated at the one end. But because there is huge amount of yolk that is present, so most of the part of the egg is covered by this yolk, and only a tiny cytoplasm is actually yolk free where the nucleus lies and where we will see that most of the divisions take place. This yolk-free cytoplasm region is basically called as the animal pole, and the opposite region is termed as the vegetal pole, and this is termed as the biopolar differentiation, which is also there in the amphibian eggs, which we have discussed. So, megalocithal and telolocithal egg, the yogi egg, and they undergo what is called as a discoidal meroblastic cleavage. So, meroblastic cleavage, we have already discussed this. The meroblastic cleavage is the incomplete cleavage, that the cleavage furrows do not pass through the yolk, and the divisions only occur in the yolk-free cytoplasm, that is the meroblastic cleavage, as against the holoblastic cleavage that occurs in the case of amphibian eggs, where the cleavage furrow passes through the complete egg and divides the complete egg into smaller cells or the blastomeres, and it is termed as a discoidal cleavage because there is a disc, as you can see, on the top of the egg, and all the divisions takes place at this yolk-free cytoplasm or this blastodisc, what it is termed as. So, the cleavage occurs in this blastodisc, a small disc of cytoplasm which is 2 to 3 mm in diameter, which is present at the yolk-free region or animal pole of the egg cell. So, here also you can see there is an animal pole, and that is the blastodisc where the embryo formation will take place. So, megalocithal, telolocithal egg undergoing discoidal and meroblastic cleavage, and after the blastula is formed, such a blastula is termed as discoblastula. So, we will talk more about it. So, let us have a look at the blastoderm stage. The first cleavage furrow in birds appears centrally in the blastodisc, and other cleavages follow to create a single layered blastoderm. So, we will just look at some pictures and we will just get to understand some more features of the avian egg, that in the first or the A part, there is a section through the yolk that reveals concentric rings of dense and white yolk. Under the blastoderm, the blastoderm is the topmost layer. Under the blastoderm, the subblastodermic space filled with white yolk forms a funnel, which is called as the latebra, that extends deep into the center of the yolk mass, where it forms a small cavity, the nucleus of Pander. So, that small cavity is termed as the nucleus of Pander. Just below the blastoderm, the funnel shaped structure is termed as the latebra, and you can see that there are concentric rings of darker and the lighter yolk which is present. So, the B part actually shows the top view. It is the cleavage in the chick embryo is meroblastic. The cleavage planes open into the surrounding yolk mass. So, you can see that there are furrows which are there, but it is only on the top, which is the blastodisc region. And the C is actually a section through the blastoderm, and the surrounding yolk reveals the subgerminal cavity and the latebra. So, the cavity between the blastoderm and the yolk mass is termed as the subgerminal cavity. That funnel shaped area is termed as the latebra, and this extends deeper into the yolk, where it forms a structure which is called as a nucleus of Pander. So, these are the certain features of the blastoderm stage as we go about it. So, this is again it shows that the blastoderm contain at least 20,000 cells. So, you can see there are two distinct areas which are there. An inner area termed as the area pellucida that can be distinguished from a more peripheral ring which is termed as the area opaca. So, you can see there is a different area pellucida and area opaca, and a marginal zone is also mentioned, and the cleavages is occurring only on the top of it, that is the blastodisc region. And if you see the section, then you can see that there is a forming cells, there is a single layered cell in the area pellucida, and it is continuous with the yolk mass lying underneath it, and then there is a marginal zone, and then there is an area opaca zone. Closest to the acellular vitelline membrane that envelopes the yolk, a simple one cell thick epithelium is continuous over both the areas, which is termed as the epiblast. So, we have already talked about the different egg membranes. So, this is right now what we are talking about, that there is a top, on the top there is a vitelline membrane. Just underneath the vitelline membrane there is a single layered cell, and this is termed as blastoderm. So, initially there is a blastodisc, and in this disc when the cells start dividing, so when the cleavage starts happening, then it attains that blastoderm stage is usually when it contains around 20,000 cells, and in the area pellucida area, there is a single layer, that is the epiblast layer, which is present, and this epiblast layer covers both the area pellucida and the area opaca. So, area pellucida and area opaca is overlaid with a single layer of cells, which is termed as the epiblast, and this entire region is termed as the blastoderm. The difference between area pellucida and area opaca, we will see just now what it is. So, this is again a pictorial representation of the same thing. So, you can see that there is an epiblast layer, the yellow colored layer which is present, and it is covering both the area pellucida as well as the area opaca and the marginal zone which is there. The area opaca, as you can see, contains several layers of large up to 150 to 200 micrometers yolky cells. Those closest to the epiblast are firmly attached to it, and this is called as the germ wall. So, it is a multi-layered area opaca, large yolky cells, very compact, and they are very adhered closely to the epiblast layer which is present, and this area opaca layer is termed as the germ wall. The area pellucida does not yet contain a continuous cell layer, but it is peppered with small islands of about 5 to 10 cells each. These are also yolky, but not as large as the deep part of the area opaca. They are around 100 micrometers in size. So, you can see that the epiblast layer is continuous. In the area opaca, there are multi-layered structure. In the area pellucida, there is only a single layer, which is the epiblast layer, and underneath it there are small islands which they are termed as hypoblast islands which are present. So, it is not a complete layer. In fact, in the area pellucida there is only a single layer, which is the epiblast layer, which is present. In between this area pellucida and area opaca, there is a marginal zone where again there are certain loose cells. There are multi-layered but little loose than what is found in the area opaca. In area opaca, they are very adhered to the epiblast, but you can see in the marginal zone they are loosely adhered and they are not completely adhered. So, between the area opaca and area pellucida is a narrow region which is known as the marginal zone. The only morphological landmark, and which is very important, is that the deep part is not strongly attached to the epiblast, unlike the area opaca. This region is known as the germ wall margin. So, germ wall is basically the multi-layered area opaca cells, and in this particular marginal zone, the morphological feature which differs is that they are loosely adhered to the epiblast layer as shown in the picture. The boundary between this area pellucida and the marginal zone is marked at the future posterior edge by a crescent shaped ridge of small cells tightly adherent to the epiblast colar sickle, also known as the robber sickle. So, colar sickle or robber sickle is very important, and you can see the location of it is basically between the area pellucida and the marginal zone. So, it is at the boundary of the area pellucida and the marginal zone. The marginal zone cells are not very much attached to the epiblast layer. They are loosely attached, but the colar sickle is again very much adhered to the epiblast layer. So, that is the region which is present basically on the posterior, the presumptive or the future posterior edge of the area pellucida and the marginal zone. And this colar sickle or robber sickle, as we will see, is the position from where the primitive streak formation occurs. So, this location and colar sickle is very important. Now, the islands which we have seen, the hypoblast islands which are present in the area pellucida, they arose by a process of what is called as polyingression or shedding that occurs throughout the area pellucida shortly before laying. So, before the egg is laid down, these small single individual cells, they just come from the epiblast, or they just polyingression occurs, or they also called it as delamination. We have seen these morphogenetic movements. So, delamination occurs and these cells start residing in the subgerminal cavity as smaller clusters, which will later fuse. So, these islands later fuse with each other to generate the primitive endodermal layer, or what is termed as the hypoblast. So, when the gastrulation starts, then these islands, they fuse together to what is called as the hypoblast or the primary hypoblast or the primary primitive endodermal layer, what it is called as. So, we will follow up the gastrulation of the avium embryo. Now, by the time a hen has laid an egg, the blastoderm contains some 20,000 cells. So, from its journey to the fertilized, that is infundibulum, when it starts moving from the infundibulum towards magnum and then to isthmus and then to the region, the cloaca region, finally it goes. During all these things, the cleavage is occurring, and by the time the egg is laid, after say 20 to 24 hours of fertilization, the blastoderm is a 20,000 cell layer thick, and rest of the divisions and the morphogenetic movements will then start when the egg is laid down. At this time, most of the cells of the area pellucida remain at the surface, forming the epiblast, while the other area pellucida cells have delaminated and migrated individually into the subgerminal cavity to form the polyinvagulation islands or primary hypoblast, which is actually an archipelago of disconnected clusters containing 5 to 10 cells each. So, you can see now these are the different stages which are shown here. So, we have talked about the first stage where there are different locations which are present, and there are two other versions which are shown, the dorsal side and the ventral side. So, nothing is happening on the dorsal side, but on the ventral side you can see there were isolated islands which were present. So, yellow color depicts the epiblast layer, and the green, smaller green circles are basically the hypoblast islands. So, you can see by the stage 12, Roman number 12, these hypoblast islands starts fusing together from posterior to anterior side, and they are forming complete green mat kind of a thing, and this is termed as the primary hypoblast. The islands of hypoblast gradually fuse together, which proceeds from the posterior to anterior to generate a continuous but relatively loose layer, which is also sometimes referred to as the hypoblast proper. So, this is how the primary hypoblast formation is taking place. Then shortly thereafter, a sheet of cells from the posterior margin of the blastoderm migrates anteriorly and pushes the primary hypoblast cells anteriorly, thereby forming the secondary hypoblast, also known as the endoblast. So, you can see that the green layer has been formed. Now, from the posterior end, those brown cells, they start moving anteriorly and they start pushing or replacing this primary hypoblast. This layer is termed as the secondary hypoblast, sickle hypoblast, or the endoblast which is there. So, you can see now the brown color is rising over the green color on the dorsal side, on the ventral side, and dorsal side there is nothing still which is happening. The resulting two-layered blastoderm, the epiblast and the hypoblast, is joined together at the marginal zone of the area opaca. The space between the two layers forms a blastocele. So, this is basically the equivalent to a blastula or a discoblastula. Now, where the two layers have been formed, the one is the epiblast layer, and now subgerminal cavity is divided into two parts. The cavity between the epiblast and the hypoblast is termed as the blastocele. The avian embryo comes entirely from the epiblast. Now, this is a very important point to be noted, that epiblast is the only layer that contribute to the cells of the embryo. The hypoblast is basically not contributing to any organ or any region of the embryo, but it forms certain extraembryonic membranes such as the yolk sac that contains the yolk that nourishes the developing embryo and the stalk of the yolk sac. But rest of these structures, the entire embryo as well as some of the other extraembryonic membranes such as the amnion and chorion, we will talk about these extraembryonic membranes in our future sessions, but that is entirely coming from the epiblast which is there. The hypoblast does not contribute any cells to the developing embryo. The hypoblast cells form portions of the external membranes, especially the yolk sac, and stalk lining the yolk mass to the endodermal digestive tube. So, those are the only regions which are formed by the hypoblast, and they will be, eventually they do not form a part of the embryo. The hypoblast cells, an important other function is that it also provide chemical signals that specify the migration of the epiblast cells. So, it does give certain kind of a stimulus or certain kind of signaling mechanism by which the epiblast cells they specify that where they have to migrate and what final positions they have to undertake, but they themselves do not form any part of the embryonic region. Now, simultaneous to the secondary hypoblast or endoblast formation, a posterior thickening called as the posterior bridge apparently derived from the collar sickle appears and the primitive streak starts to form immediately after. So, these two things simultaneously occur. That first is the formation of the hypoblast layer. We have seen that how the primary and the secondary hypoblast is formed. Then simultaneously on the dorsal side we can visualize that there is a kind of a thickening or a posterior ridge starts appearing, and this is basically termed as the primitive streak that is occurring. Now, the cell interactions leading to the primitive streak formation, there are three main sources that have been proposed to influence the primitive streak formation. First is the hypoblast or the endoblast, second is the collar sickle, and the third is the posterior marginal zone. So, all the three cells in these particular locations, the constituent of these three regions, the hypoblast layer, the collar sickle layer, and the posterior marginal zone area, these three cells contribute to the formation of the primitive streak which is there. So, now we will follow up the formation of the primitive streak and how it elongates. The initial appearance of the streak is extremely rapid. The embryo goes from having no visible axial structures to developing a triangular dense streak in about 30 minutes, suggesting that primitive streak initiation is accompanied by massive ingression. So, how is this primitive streak formed? Location, now we know that it basically starts from the posterior edge of the marginal zone at the location of the collar sickle, that is where the position of the primitive streak formation where it starts from. And how does that occur? It's a very, very rapid phenomena. Therefore, they say that it is accompanied by massive ingression of the cells that is occurring. The early triangular streak, which is seen in stage 2, is made up of a dense accumulation of middle layer cells between the epiblast and the endoblast. So, you can see there is an epiblast which is there and there is an endoblast which is there, and then this middle layer at the position of the collar sickle, this middle layer is formed, which can be visualized now from the dorsal side also. On the ventral side, again this is the hypoblast layer which has been displaced by the secondary endoblast layer, and in this particular area this is the ridge which is triangular ridge which is forming. It rapidly, so it appears as a ridge or it appears as a thickening, and then soon it straightens up and to become a mesenchymal rod of parallel sides. So, we will see that this primitive streak that appears as this kind of a hilly region will become straighten up, and we will see that it will cover the 3/4 of this particular layer. At this stage, there is still no groove in the overlying epiblast. So, there are it's kind of a rod, but there is no emptiness, so there is no groove that has been formed, and the basement membrane is largely intact. So, there is no place from where the cells can enter inside. That groove hasn't been formed, but the structure of the primitive streak has been laid. The streak is first visible as cells that accumulate in the middle layer, followed by a thickening of the epiblast at the posterior marginal zone. So, that is how the primitive streak starts formation. Elongation occurs because this thickening is initiated. So, how does this thickening initiate? This thickening is initiated by the increase in the height of the cells forming the center of the primitive streak. The presumptive streak cells that surround them become globular and motile. So, they start digesting the extracellular matrix, they become mobile, and because of this mobile they undergo what is called as the mediolateral extension, and they start moving in that manner, and this convergent extension then makes them become the makes the primitive streak elongate. So, the thickening occurs because the cells becomes increased in size, and around that central cells, the cells become motile, start digesting the ECM, and then starts moving in the anterior direction to form to elongate the primitive streak. This process allows their intercalation, which is mediolaterally, and convergent extension, which is responsible for the progression of the streak. So, initiation of the primitive streak is by the thickening of the central cells at the location of the colar sickle at the posterior marginal zone. So, that kind of thickening starts occurring, and the cells surrounding it, they become motile and they start intercalating mediolaterally, several layers, when then they tend to become a single layered, and therefore the elongation of the primitive streak occurs. Soon afterwards, two processes takes place more or less simultaneously. The appearance of a longitudinal groove. So, by this time there was no groove, or there was no empty hollowness in the streak, but after this primitive groove forms. This primitive groove is actually the area from where the cells will, that will kind of create a space from where the adjacent cells will keep on going inside. And the second thing is the formation of the Hensons Nord, that is a bulge or thickening in the anterior-most portion of the developing primitive streak. So, the appearance of a longitudinal groove in the epiblast overlying the streak and the start of the lateral migration of the mesenchyme of the streak at right angles to the axis of the streak to establish the lateral plate. This process defines what is termed as the stage 3+. So, in the development of avian embryos, different stages are numbered initially by the Roman numerals, and then by 2, 3, 3+, and different stages are numbered, and there are different different classification of these stages or nomenclature for these stages. And so, we are following right now the stage which is shown here, the numbering which is shown here. So, these processes define the stage 3+. So, here now you can see that the primitive streak had started formation, and now you can see that that hilly region has actually become elongated. So, the central proportion become thick, and now the surrounding cells, they keep on converging, they keep on converging extension. So, they keep on converging and they keep on extending, and therefore, now it has become elongated. And on the ventral side, now you can see that this kind of appearance has started coming. So, the groove has been laid inside it, the cells will just go inside and they will come out at their right angles. So, these are the lateral plate mesodermal cells, and at the anterior most portion from where the cells are going inside, they will now start displacing the endoblast or the secondary hypoblast, and these cells are actually the definitive endodermal cells. So, this green region is actually the green cells are the endodermal cells which have been migrating from the epiblast, going into the deep, so much deep that it is displacing the hypoblast cells from this particular area. The primitive groove then serves as an opening through which the migrating cells pass into the blastocele, and therefore primitive groove is analogous to the amphibian blastopore. So, that primitive groove is basically from where the cells will go inside, and therefore it is the equivalent to the animal blastopore, an amphibian blastopore. The stage 4 is marked by the appearance of a distinct bulge at the top of the streak encompassing all the 3 layers, and this is termed as the Hensons Nord or the primitive node. The center of this node contains a funnel shaped depression which is termed as the primitive pit through which the cells can pass into the blastocele. Hensons Nord is the functional equivalent of the dorsal lip of the blastopore or the amphibian blastopore. 2 different population of cells form the Hensons Nord. The one cell population called as the posterior cells resides deep to the epiblast at the midpoint of the colar sickle. It remains at this position until the primitive streak starts to form and then moves anteriorly with the tip of the advancing streak. And the second population of cells are called as the central cell. They resides in the epiblast during these early stages. The movements almost immediately move these cells to the middle of the blastoderm where these movements stop. So, less than 2 hours elapse between the early short streak at stage 2 and the almost fully elongated stage 3 streak which is there. So, you can see in the diagrams that after this there is a stage 4 plus that occurs, a small triangular mass. So, this is where the primitive streak started forming. this is how it started elongating, and this is the fully developed primitive streak which is having the Hensons node at the top, and you can see the fate map also. So, we will talk about now the mechanism of gastrulation. So, you can see the, which region will form this. So the anterior portion will give rise to the cells will move inside and will give rise to the notochord. So, this is the epidermal ectoderm, the neural ectoderm, the paraxilium mesoderm, the lateral plate mesoderm, and the extraembryonic mesoderm. So, after this 3 plus, now the Henson node had been formed, and you will then see that there is a emerging tip of the head process which is there that contain the precursors for the precaudal mesendoderm. At this time, the future neural plate starts to become morphologically and molecular distinct, and that is termed as then the neurula stage which is there. So, the primitive streak has now been formed and elongated, and then we can follow the later mechanism of gastrulation in future sessions. Thank you. In this particular session, we will follow up the formation of the 3 germ layers in birds, and we will also understand the chick organizer. So, we have seen that the endoblast layer, the primitive streak formation has taken place, the primitive streak has formed, the primitive groove has formed, and now the cells have to migrate so that the formation of the germ layers can occur. So, to start with the mechanism of gastrulation, how it occurs, the first thing, an important thing is to study the fate map, what future positions they have to acquire. So, you can look at the fate maps and cell movements at the Blastoderm stage. So, we already discussed this 10, 11 and 13 stage. So, at 10, 11 and 13 stage, you can see the position of the precaudal, the notochord, the medial somite. So, these are all the mesodermal regions which are there, and then the neural ectoderm, the forebrain, midbrain and hindbrain, and the spinal cord which is there. This is the white portion is the gut endoderm, and the yellow one we have already talked about is basically the epiblast layer which is there. The future dorsal tissues, which are basically the prospective organizer and its derivatives, the endodermal, precaudal, mesoderm and the notochord, they are found just central to and adjacent to the coelursicle. So, we have already seen that this is the posterior marginal zone, and this is where the coelursicle was present. And you can see that all this precaudal, notochord and the medial somite, so all these regions are actually present in the central portion at the 10th stage when the primitive streak had just started to form. And similarly, you can see that the forebrain, midbrain and hindbrain, the neural ectoderm region is also just here which is present. These territories quickly move towards the center of the blastoderm, and they gradually become replaced in their original portion by more lateral regions of epiblast, the progressively more ventral fates like somite, intermediate mesoderm, etc. So, they will start moving towards the center and they will start moving towards the anterior, and as you can see that this particular region is then replaced mostly by the lateral plate, more ventral portion basically which is there. So, they get replaced by such areas. The posterior margin of the area pellucida contains a bilateral gradation of dorsal to ventral fates, dorsal at the posterior midpoint. So, this is basically the most dorsal region, and then when they start migrating, as you can see, they will start, it's kind of a dorsoventral axis which will be formed. The subsequent movements fold this arrangement so that the most dorsal fates become located most anteriorly along this line. So, to start with, this is the dorsal region which is there, and then they will fold and they will start moving anteriorly, and you can see that by this stage most dorsal regions are in the most anterior portion, and these are the lateral plate mesoderms, and then the ventral portion which will be on the posterior edge that will be remain. So, it is kind of a folding that has occurred, and this depicts how the cells are moving. So, this is the fate map, and let us follow how this happens. So, the migration through the primitive streak, we will start with the formation of the endoderm and the mesoderm. The first cells to migrate through the Hensons node are those destined to become the pharyngeal endoderm of the foregut. So, Hensons node is also equivalent to what is termed as the dorsal lip of the blastopore. So, as in amphibian gastrulation, through the dorsal lip of blastopore, the first cells to move inside were the pharyngeal endoderm of the foregut, and the same thing happens in case of chick, that the first cells to migrate through the Hensons node is basically the pharyngeal endoderm of the foregut. These endodermal cells migrate anteriorly and they eventually displace the hypoblast cells, causing the hypoblast cells. So, they will start moving from the Hensons node and they will start moving anteriorly, and when they start moving anteriorly, because they are the endodermal layer, they will go as deep as where the hypoblast layer was present or the endoblast layer was present, and they will start displacing the endoblast layer towards the anterior region, and then this endoblast layer will be actually confined to a very small region on the anterior portion of the area pellucida in the shape of a crescent, and that is termed as the germinal crescent. And if you recall from the gametogenesis, that this germinal crescent is from where the germ cells will arise and they will migrate to the developing gonad to give rise to the gametes. Depending upon whether the gonad is a male gonad or a female gonad, it will develop and proliferate and divide to form the male gamete or the female gamete which is there. The next cells entering the blastocele through Hensons node also move anteriorly, but they do not move as far ventrally as the presumptive foregut endodermal cells. Rather, they remain between the endoderm and the epiblast to form the mesenchyme, the head mesenchyme and the precaudal plate mesoderm. So, the pharyngeal endoderm move as deep as where the hypoblast layer was present. The second cells to go inside is again the head mesoderm or the precaudal mesoderm, the cauda mesoderm, but the head mesoderm as well as the cauda mesoderm, they do not go as deep as the endodermal layer, but they will start lying in between the developing endodermal layer and the epiblast, and they form the mesodermal cells. These early ingressing cells also move anteriorly, pushing up the anterior midline region of the epiblast to term what is called as the head process. So, you can see this head process has been developed. So, this is the Hensons node and they have moved anteriorly. The first is the pharyngeal endoderm which has started developing, and then the head process region has been developing. The next cells migrating through Hensons node thus becomes the cauda mesodermal. So, pharyngeal endoderm, head mesoderm followed by the cauda mesodermal cells. These cells extend up to the presumptive midbrain where they meet the precaudal plate. The hindbrain and trunk forms from the cauda mesoderm at the level of Hensons node and caudal to head. Meanwhile, the cells continue migrating inwardly through the lateral position of the primitive streak. These cells separate into two layers, the deeper layer that joins the hypoblast along its midline and displaces the hypoblast cells to the sides. These deep moving cells give rise to the endodermal organs of the embryo as well as most of the extraembryonic membranes. The second migrating layer spreads between this endoderm and the epiblast, forming a loose layer of cells, and these middle layer cells generate the mesodermal portion of the embryo and the extraembryonic membrane. So, what is happening anteriorly is also happening laterally. So, once the sides of the primitive streak from where the lateral migration is occurring, then that lateral migration, the one layer goes deep inside and displaces the hypoblast and they will form the endodermal organ. Another layer goes inside but doesn't go so much deep and lies in between the epiblast and the endodermal layer, and that forms the lateral mesoderm and give rise to the mesodermal structures and most of the extraembryonic membranes which is there. The long axis of the primitive streak does not correspond to the future head-tail axis of the embryo, but rather to the future dorsoventral axis of the mesoderm. The anterior streak give rise to the most dorsal axial structure with more ventral structure arising from progressively more posterior streak positions which are there. So, this diagram picture again shows the kind of movements that is happening in the anterior portion of the primitive streak. This is a section just to show that at the anterior most portion the cells are going inside and going anteriorly. These are the pharyngeal endodermal cells. These cells which are going inside and going towards this side is the cauda mesodermal cells, and these are the lateral mesodermal cells which are there. And when you cut a section, that you can see that some of the cells are migrating deep inside and displacing the secondary hypoblast cells. These are termed as the endodermal cells. Some cells are moving inside, but they are not going so much deep, and they start lying in between this blue layer which is the epiblast layer and this orange layer which is the endodermal layer, and this layer basically forms the mesodermal layer that is there. And this again shows the fate map. Then the one which is going most anterior forms the notochord or the precaudal cells. The ones which is going to the anterior but not as much anterior as from the Hensons Nord, then they form the medial somoid, lateral somoid, they form the kidney and the lateral plate, and this is. So, these are basically all the mesodermal cells, and based upon the location from the dorsal to the ventral region, they are telling us that they will give rise to which part of the in the future embryo. So, the most anterior one will give rise to the precaudal, these are the cauda mesoderm, then comes the somoid, lateral somoid, kidney, lateral plate, mesodermal cells, etc. And the one which is going inside from the most posterior part of the primitive streak is giving rise to the extra embryonic mesoderm which is there. So, this again depicts that this is the head process that has been formed, as you can see, and this is how the even the somites have been started and the neurulation has started here, and then you can further follow the fate map that now this is the primitive streak and the central portion or the notochord portion, and this is basically the neural ectoderm, this is the epidermal ectoderm, and these are the mesodermal cells which are there, which have now formed in this particular way. Now, reviewing again the general morphology and movements separately, how the ectodermal, endodermal and mesodermal layers are moving. The first is the general morphology and movements of the upper layer or the epiblast of the chick embryo at stages from the 10th stage when it is laid to the 5th stage when the early neurulation is starting. So, you can see that this is the anterior and this is the posterior. This is the posterior marginal zone where the colar sickle was present. So, looking at what are the changes occurring in the epiblast layer. So, this is the area pellucida which was there. These are the islands which are there. These islands starts fusing. They start forming this hypoblast layer. So, this is the hypoblast layer that has been formed on the ventral side. On the dorsal side, you can see that the primitive streak has started in the form of a ridge of a thickening at this posterior portion. This is the stage 2. So, at stage 3, this primitive streak, there is a convergent extension that is occurring, and this primitive structure keeps on elongating. You can see now this is keeping on elongating. By stage 4, there is a Hensons Nord that has developed, the dorsal lip of blastopore, and this dorsal lip of blastopore, the cells will start moving and they will give rise to the head process, as can be seen here in the stage number 5, where then eventually the neurulation will start. The marginal zone is shown in pale pink, the colar sickle is shown in red, the upper layer movements are indicated by blue arrows. In this figure, the stages of development before the primitive streak formation follow Ayal-Giladi and Koichev and are shown in Roman numerals, and those after the appearance of the primitive streak follow Hamburger and Hamilton and appear as Arabic numbers. So, this is the nomenclature of basically the stages which is formed in this case. So, this is how the epiblast, the movement occurs in the epiblast and how it is there. Then we will follow up the different components of hypoblast, endoblast and endoderm of the lower layer of the area pellucida of the chick embryo and their movements. So, how are they forming now? So, you can see that again this is the region where these are basically the hypoblast islands. So, they will start formation. The hypoblast islands have been fused together, and these hypoblast islands are now giving a particular layer to the primary hypoblast. Now you can see this primary hypoblast is displaced by the secondary hypoblast. So this green is basically the secondary hypoblast which has occurred. Now at stage 2, this is the secondary hypoblast and this is the thickening which has occurred. Now this thickening has started elongating, and after elongation you can see that the first cells which start moving is the pharyngeal endoderm. So this is basically the endodermal layer which is starting formation. So this endodermal layer, the first one, the pharyngeal endoderm, will go towards the anterior edge and they will displace this green region and they will confine it to this kind of an area which is called as the grey crescent area, or basically the secondary hypoblast. So this area is basically the green, the germinal crescent, and this endoblast layer, now they are going from the anterior portion Hensons node, and then they will subsequently start migrating in the lateral regions also. And now you can see that the endodermal layer has completely replaced, is going to replace the entire secondary hypoblast which is there. And this secondary hypoblast or endoblast is basically confined to only this anterior portion which is termed as the germinal crescent. So hypoblast is shown in the orange, the endoblast in green, and the endoderm in pale blue. From stage 3 plus, the hypoblast becomes confined to a secondary shaped region, to a crescent shaped region at the anterior end of the embryo, which also contains the germ cells. This region is therefore called as the germinal crescent. So you can see this is the germinal crescent, and this green secondary hypoblast area is actually will be displaced by this developing endodermal layer. So this is how the endodermal layer is formed. Then we will follow the middle layer or the mesodermal layer, what happens and how that moves. So you can again see this is the colar sickle, and the middle layer will basically start migrating, or the mesodermal cells will start migrating from the lateral edges. So while this primitive streak formation is occurring, so you can see this is the primitive streak, and this primitive streak, by the time they are increasing, this lateral plate mesoderm, they will go and they will not go as deep as the endodermal layer, and now you can see that this mesodermal layer is being laid. After laying here, then this chorda mesodermal cells are moving towards the anterior side. So the lateral movement starts first and the chorda mesodermal layers will start at the later end, and this chorda mesodermal cells will give rise to the head process which has been forming, and this is the lateral plate mesoderm which is moving. So colar sickle is shown in red here, the marginal zone in pale pink, and the mesoderm is in the green. The mesoderm movements are indicated by the blue arrows. So this is how the mesodermal movement occurs. So we have seen now what happens to the epiblast layer, what happens to the endoblast layer or the endodermal layer, how they are formed, and then what happens to basically the mesodermal layer or how the mesoderm is laid down in the chick embryo. So these are the prominent gastrulation movements. After the formation of these three layers, the regression of the primitive streak starts. So regression of the primitive, while mesodermal ingression continues, the primitive streak starts to regress, moving the Hensons node from near the center of the area pellucida to a more posterior position. So while this displaced mesodermal ingression is occurring, and it is displacing forming the lateral plate mesoderm etc. and different mesodermal somites, this Hensons node will start regressing, and as you can see, by the time the head fold is forming and by the time the somite starts appearing, that the Hensons node has almost reached the posterior most portion. So this much is the length of the primitive streak that has been there. As the node moves posteriorly, the notochord is laid down starting at the level of the future midbrain. So by the time this is there, the notochord will start laying down, and after this stage the neural ectoderm will also, the neurulation will also start. But the regression of the primitive streak is then an important phenomena, because by this time all the three cells of all the three layers have acquired their positions in the region. Then finally Hensons node regresses to its most posterior portion, forming the anal region. So this opening, the blastopore, just like in amphibians, this will go and lie as an opening at the posterior end and will become the anal region. So this is how the endoderm and mesoderm layers are formed. The ectodermal layer is essentially by epiboly, like in case of amphibian gastrulation. The ectodermal cells migrate to surround the yolk by the epibole. It involves the continuous production of new cellular material and the migration of the presumptive ectodermal cells along the underside of the vitelline envelope. So, the continuous production of new cellular material, so the divisions are continuous, and the presumptive ectodermal cells, they move along the underside of the vitelline envelope to encompass the entire yolk mass and to cover the entire underlying layers which have been forming, and that is basically the ectodermal layer which is there. So, as avian gastrulation draws to a close, the ectoderm has surrounding the yolk, the endoderm has replaced the hypoblast or the endoblast, and the mesoderm has positioned itself between these two regions, and therefore the three germ layers have been formed in case of avian. So, this is how the formation of the germ layers occur. Now, here only I will talk about a little bit about the organizer region. So, we have seen in the case of amphibians how the embryonic induction occurs and how the dorsal lip of blastopore act as an organizer. Similarly, in chick also we have an organizer region that basically organizes the entire tissues to form the future organs which are there. So, there is an induction that is occurring and there is an organizer region which is there. So, the chick organizer is forms just anteriorly to the post marginal zone. So, here you can see this is again showing the same area. This is the epiblast layer, this is the developing hypoblast, this is the colar sickle, and this is the deep layer of the marginal zone, and this is the area opaca which is there. So, epiblast and the middle layer cells in the anterior portion of the colar sickle becomes the Hensons node. So, we talked about the two kind of cells which form the Hensons node, the deep cells and the central cells. So, Hensons node are basically formed from these cells. Eventually they will move in the front and they will form the Hensons node, and this is the position where the chick organizer is residing. Posterior portion of contributes to the posterior portion of the primitive streak. So, Hensons node has long been known to be the avian equivalent of the amphibian dorsal blastopore lip, since it is. So, we have talked about that this Hensons node is equivalent to the dorsal lip of blastopore as in case of amphibians, and what was the role of the blastopore? That it is the site where the gastrulation begins. So, actually the morphogenetic movements also start. So, the primitive streak formation occurs, but the groove does not develop until it reaches almost the center, and then when the central cells forms this Hensons node, the central deeper cells and the surrounding cells form the Hensons node, and the groove develops, then the first movement or the gastrulation movements, the pharyngeal endodermal movements occurs through this Hensons node. So, this is very important structure that the first gastrulation starts from at this point, then the region whose cells become the chorda mesodermal cells. So, it is an important part that the pharyngeal endodermal cells move from this Hensons node towards the anterior region and forms the lower endodermal cells, then the region whose cells become the chorda mesodermal cells. So, we have seen that the dorsal blastopore lip also, the first cells to go over the bottleneck cells which were forming the roof of the archentron, which were forming in the amphibian gastrulation, the cells to move inside was the chorda mesodermal cells which was forming the dorsal lip of blastopore, and similarly in Hensons node the chorda mesodermal cells forms the region becomes the chorda mesodermal cells. Then the region is called as the organizer because the cells can organize a secondary embryonic axis when transplanted into other locations of the gastrula. So, this was an experiment done by Waddington where actually this is the duck embryo and this is the chick embryo, and you can visualize both the embryos because of different pigmentation. So, you transplanted this duck Hensons node at a position when the primitive streak was forming, and next to it you just transplant this, and then you can see that a new neural tube is induced. So this is termed as an organizer because it has the ability and potential, the most important property of being an organizer, that it has the ability or potential to organize the tissues surrounding it to become the neural tube. Otherwise, this particular region will not give rise to, there will be no neural axis that is formed here. This is the normal thing which is going to develop. This is the primitive streak has regressed here, these are the somites, this is the head process which has been developed. So this is the host embryo, but after the transplantation of this Hensons node, you can see that there is another axis that has been started and the neural tube induction has taken place. So, and Hensons node is an important structure and it basically gives rise to, or it helps in the organization of the tissue embryo, and therefore it is termed as the chick organizer region. So, we have seen the avian development, we followed it through the type of egg. It has the kind of cleavage it occurs. We haven't gone into detail the fertilization mechanism, but we know that the fertilization occurs in one part of the oviduct, which is termed as the infundibulum, the funnel shaped structure. The fertilization occurs there, and by the time the fertilized egg is laid, there is a division in the blastodisc which is termed as the blastoderm, and this blastoderm then give rise to there are regions at the posterior end of the blastoderm or the posterior marginal zone which is termed as the location from where the primitive streak will start forming. So that is basically termed as the equivalent to the blastopore, and the Hensons node, which is the dorsal lip of the blastopore, is the organizer region in the chick embryo. That is from where the gastrulation or the morphogenetic movements will start. The main type of morphogenetic movements that occur in the chick is the polyingression that has been occurring, the delamination when initially the hypoblast islands have been formed and they are kind of a splitting of a layer, epiblast layer that has occurred. The mesodermal and everything moves by the ingression and the epiboly of the ectoderm. So, ingression, delamination and epiboly are the major morphogenetic movements that occur in case of the chick embryo. So, that is how the chick develops up to the formation, up to the gastrulation we have followed, and further we will follow in the future sessions.
On that note, I would like to thank ma'am for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.